Hello, hi. Welcome back to the next lecture. In this lecture of Principle of Inheritance and Variation or Genetics series, in this lecture we will be dealing about a small topic, a small two topics, which is the monohybrid cross and also the Mendelism extension. What is actually Mendelism? Mendelism are some principle which is proposed or which are experimentally concluded by Mendel in 1800s or 1860s. Okay, you know Mendel, Grigor Johann Mendel. He was a scientist. He was also a preacher. He has done a variety experiments on different organisms to conclude about some principles about genetics, how or why there is a certain characteral difference or why there is a similarities between siblings, yes, right, or why there is a variation between siblings. I think you understood. It is a progeny from a parent, different progeny from a single parent, two types of parents, right? So why there is a variation or why there is a inheritance? To understand it very clearly, Grigor Johann Mendel has made a different of experiments. I have conducted a different of various experiments from which he concluded certain laws. The main two laws from his experiment, first experiment was principle of dominance and principle of segregation. So we will be studying his first experiment. First experiment means he has done a variety of experiments. We will study a one experiment from that, which is monohybrid cross. In this lecture, we will be dealing about monohybrid cross. Okay, we will deal about principle of dominance, principle of segregation, which are certain laws or principle which he concluded from this experiment. And how did he done this cross? How did he done this cross, right? And also in the coming part, we will also deal with the extension of Mendelism. Apart from the things Mendel have done, there is something extra things also we can understand from Mendelism or Mendel's experiments. So I will say some points regarding Mendelism. Okay, so Grigor Johann Mendel was the scientist who experimented in this hybrid cross, right? So in case of Mendelian genetics or classical genetics, what is Mendelian genetics or classical genetics? It is the genetics which is the previous or old genetics, right? There are mainly many variation from his genetics when we are up to date. There are many different variation from the things what Mendel have done. Okay, so Mendelian genetics is applicable in case of sexually reproducing organism. Mendelian genetics is applicable in case of sexually reproducing organism like human beings or any other sexually reproducing organism like plants, etc. Why Mendelian genetics is applicable in case of sexually reproducing organism? Because sexually reproducing organism produce gametes, produce haploid gametes, and these two haploid gametes, one from parent 1, one from parent 2, considered as a father and mother, they fuse together to form a zygote which is a diploid. So just consider a gamete from a different parent and a different parent is fusing together to form a different an organism with two different gametes, right? So there is a fusion. This is the main reason for Mendelian genetics to be applicable in case of sexually reproducing organism. It is not applicable in case of viruses and bacteria. Next point is that Mendelian genetics is based on qualitative character. What do you mean by qualitative character? Qualitative character, just I think you have the basic idea of genetics, right? So I will be saying that Mendelian genetics is based on qualitative inheritance or qualitative character. He only take qualitative character. What is that meaning? It means that, Mendel have experimented on garden pea, right? Paisam sativa, garden pea. I will say the reasons why he choose garden pea. He have also done many experiments in honeybees and certain other organism also. But why did he took garden pea as a great example, as a model organism? I will say it later. Now I was saying about the qualitative character. So Mendel took Pisum sativum, right? A pea plant. The pea plant Mendel took was only having two contrasting character. Two contrasting type for a single character. Example, if he took the height of a Pisum sativum plant, there was only tall height plant and there was only dwarf plant. There was no intermediate. This is why I said that he only consider his experiment was only based on qualitative character. If I draw a graph, just check. I am just drawing a graph. Okay. Here, this is x-axis. This is y-axis. Just it is not needed even though. So this is plant 1 and this is plant 2. Plant 2. Let me draw this plant 1/4 length. Okay. Just consider this example. These two plants, plant 1 and plant 2. So there are only two different contrasting types. One is dwarf and one is tall. So there is no intermediate stage. There is no middle stage, right? There is no intermediate stage. Usually the plant Pisum sativum tall height was like 4 meter and this was half meter. Okay, so this is the case of his experiment. His experiment was based on qualitative character or discontinuous characters. There is only either a tall parent or a short parent. There is no intermediate character. So this is the point number 2. And point number 3 to be remembered is that Mendelian genetics was based strictly on inheritance, genetic inheritance. He did not involve any other extra environmental variation or any other variation. He did not consider an environmental variation. So this is the third point. You know why? He did not consider, we are dealing about Mendelian genetics, and he did not consider environmental characters. If you just look outside, there are certain plants which grow according to the environment, right? Even though they are having some genetic inheritance, but environment also plays a role. If the amount of water is less, even though the plant will grow in a good height, if the water is less or any other mineral are less, then the plant cannot grow to a certain height. There is a limit up to which a plant can grow. But to reach that limit, there are certain environmental characters which can constitute or which constitute everything to that plant to reach a certain height. In case of Mendelian genetics, he did not involve any environmental character. The next point to be remembered is that for Mendel's, each character have a contrasting pair type, which means each character has a contrasting pair or each character have two alternative forms, which means I am taking the height of a plant or height of a Pisum sativum. So the plant will be having a tall and a dwarf. If I am taking the flower position of that plant, Pisum sativum, the plant will be either axile or in the terminal position. the plant will be the flower will be either in the axile position or the flower will be either in the terminal position. If you know, in Mendel's experiment, he took about 7 contrasting character. 7 contrasting character. So one of the character is the stem height. Next is the stem. One of the characters is the stem height or the height of the plant. Next is the flower position. Next is the flower color. Next is the seed shape. Next is the seed color. There are 2 other also, the pod color and the pod shape. 7 contrasting characters was taken by Mendel. You know, I will show it here. Okay. Look, these were the 7 contrasting characters which were taken by Mendel. It is given in your textbook, table 4.1. If you open textbook with you, it will be better. Always read textbook. Textbook is very easy to be very easy to be read. It's very understanding. So please read the textbook. If you read it, you will understand it. So these are the 7 contrasting character. There are contrasting character. Look, contrasting means it also have another form. So we were dealing about the monohybrid cross. We were talking about Mendel's experiment character. Okay. The next character to be remembered, he did not include any environmental factor. There was 2 alternative forms for a single character, right? So that was the thing, and you should understand what were the 7 characters which were taken by Mendel. Just understand like this. A tall plant, what is it? A tall plant with axial flower, a tall plant with violet axial flower, a tall plant with violet axial flower having inflated green pods with yellow round seeds. The statement I have said right now have the 7 characters which are dominant. I will say it once more. A tall plant with axial violet flowers containing pods containing an inflated green pods having a yellow round seeds. These are the 7 characters which were taken by Mendel for his experiment. These 7 characters are the characters of Pisum sativum, which is known as pea plant, a common pea plant. I think I have the picture of the plant. You may have seen it. Okay, look, this is the plant, Pisum sativum. Look how beautiful it is, right? This was the garden pea which Mendel has taken for his experiments. Gregor Johann Mendel, right? He was a good mathematician. He was a good priest. He was a good biologist, right? So some of the characters, these are the characters he took to study, right? Look, seed endosperm. Actually, seed color means it is the color of the endosperm, not the coat color. The coat color is actually gray and white. Just actually it is not needed in textbook. Only just understand textbook, because these are some extra things. And you just look the pod color and the pod shape, right? Pod shape, I actually said that it is constructed and inflated. You know both the meaning. Pod color. I have said the pod color is green. Also there is yellow. Green color, which it means it is dominant in nature. Okay. You know what is dominant and what is recessive. I will once more explain while we are doing the experiment. While we are conducting the experiment, we will conduct Mendel's experiment once more. Okay. So this is the condition of the flower position and flower position. Look. So we just check the flower position. We have 2 conditions of flower position. One is the axial flower position and terminal flower position. What is axial? Axial means it is present in the axial branch. It is present in the branch. The flower, if it is present in the branch, it is known as axial, and if it is present in the terminal or it is present at the stem, it is known as terminal flowering. This is the case of the Mendel's case. So these are the things to be noted. I earlier said that I will not be giving any notes. I will repeat the points and you should write down the notes, because in this online mode it is very difficult to write it in the board. Next, okay, so we were dealing about principle, Mendelian some characters of the Mendel some character. So the garden pea, the scientific name of garden pea is Pisum sativum. Actually, we know the rule for writing a scientific name. Okay, I am writing it in my computer, so I can give it in italics form. Look, I have give it in italics. This is italics. This is without italics. This is with italics, right? So there is no need for me to underline both the genera and the species name. If I am writing without italics, then I should just give underline. This is the common rule for writing the scientific name. Why there is a rule? Because if we are writing it in a person can't understand whether it is a normal word or it is a scientific name of some organism. So if we underline it, giving a space, we can understand it is a scientific name. And usually the genera or the species name should be written. The first alphabet should be in small letters. This is the case. So this is the experiment, as this was the scientific name of the garden pea which he took for his experiments. Also, these organisms are diploid in nature, right? So it is having a diploid condition. 2n is equal to 14, 1 4, 2n is equal to 1 4, 2n is equal to 1 4 means it is having 7 chromosome, right? 7 chromosome and 7 pair of chromosome. It is having 7 chromosome, or we can say that it is having a 7 pair. 7 into 2, 14. So a total of 14 chromosome. This is a pair. It will be having 7 pair. 1, 2, 3, 4, 5. Or we can count it as 1, 2, 3, 4, 5, 6, 7, 8. Up to 14. So there are 7 chromosomes. This is the case of Pisum sativum, a pea plant. I have earlier said I have shown the diagram, right? So these are the cases of Mendel's experiment. I think there is no need to repeat it again, because if you want to repeat, please go back 5-10 seconds. You can understand the things. Go back from, if you want to write the notes, okay, you can go back and write the notes, or pause it, write the notes. It is according to you. You can do. Next. So I have said something about Mendel's, just basic idea of Mendel's experiment, right? He has done, Mendel has done an experiment for 10 years of experiment. 10 years conducting experiments in garden peas and certain other organism also. He was such patient, and he was having such patience, right? And he was not in a much hurry to complete his experiments and to submit. He just do it without a tension. So there is no tension required for anything. Just relax. But if there is a time period, you should make a well-disciplined plan for it, right? To complete it, anything. So we are now, okay. In 1865, Mendel submitted his paper in Broome Society for Natural Science. His paper name was Plant Hybridization, Experiments on Plant Hybridization. Paper means he have conducted certain experiments and he have concluded it with a result that is known as paper. So he submitted his paper in 1865 to Broome Society of Natural Science, and in the next year, in 1866, it got published. But it took 40 years to accept his result in science societies, because it was hidden 40 years. No one was knowing about it. 1900s, in the beginning of 1900s, only the people get to know, or the scientific society gets to know about this experiment, because people were not knowing about how characters were inherited, how progenies were having same characters of their parents, or there are some variation from their parents, it is because they were not aware, the scientific society, or they weren't aware, because during Mendel's period also there were several other scientists who were involved in these types of experiments, but no one was able to give a clear definition of how these characters were passed down. There are many more details to be said. If I am saying this, we will not be getting the time, so I will say in between the class, and I think you will enjoy. So now we can do the cross, monohybrid cross, what Mendel have done. So we are going to the most interesting experiment, right? So okay, I have written it earlier. Okay, just consider. Okay, let us start our experiment. So we are going to take two parents. We are studying about, first is about the monohybrid cross. There is also other cross which we will be learning it in the next part, or next part 3. Okay, in this lecture we will be dealing about monohybrid cross. What is monohybrid? What is monohybrid cross? Actually, mono means one, we know that, right? Hybrid, what is hybrid? Hybrid is actually the intermixed form, or yes, it is the intermixed form of two different things, right? That is actually a hybrid. Okay, there are certain characters from both of the part or parents. So in a monohybrid cross, in Mendel's monohybrid cross, he took only one character. He only took one character in his experiment. In dihybrid cross, he have taken two character. If in a trihybrid cross, he have taken three character. In a tetrahybrid cross, or a pentahybrid cross, or a hexahybrid cross, he have taken 4, 5, 6 genes, etc. Or 6 character. You know that a character codes for a gene codes for a character in this experiment. Okay, there are changes. We will be studying the changes, okay, in the later parts, but here just consider a gene codes for a character. Example, in monohybrid cross, if Mendel took tall height as a character, we know that tall height, or the if Mendel have taken height as a character, we know that there are two alternative forms for height: dwarf height and a tall height. Dwarf height means the plant will be small. The tall height means the plant will be very tall. So either he can take either he can take height, or he can take flower position, or he can take flower color, or he can take pod size, or he can take the seed shape, or he can take the seed color, or pod color, etc. So 7 different character, but he have taken every different character, and he have taken every different character and conducted the experiment in monohybrid case. For our ease of understanding, we will be taking tall height as a character. Okay, we can take any other characters also, that is according to our wish. I think in your textbook height has been taken. If you want, we will do the experiment with other character also, or you can do the character after we do this experiment, right? So we are taking, in this experiment, we are taking height as the character for monohybrid cross. Okay, I think you have understood it very clearly. So we can do the cross. So parents, you know, in Mendel has done this experiment in a certain manner. There is no question about asking why he have done that, why he didn't do that, because it is our understanding, right? Mendel has done his experiment in his ways, so he have concluded certain result in his ways. So we are following that way first, and we will make some correction in the last end part of this chapter. Chapter means inheritance of variation. So first we will understand what Mendel have done. Okay, so there is a parent generation, there is a parent condition. So parent 1 and parent 2. Consider parent 1 as a male, parent 2 as a female. You know, this is the sign for female and this is the sign for male. You know, this is understand. Okay, you can convert it also according to your wish. So he have taken parent 1 and he has taken parent 2. And we need to understand that in Mendel's cross, parent generation are homozygous in nature. It is pure homozygous in nature. So he had took the pure homozygous dominant parent. Here he have taken a pure homozygous recessive parent. This was his experiment. He have done this experiment according to this. So we are taking the height tall, right? Let me take my pen. So he have taken the height of the plant. He have taken the height. So parent 1 is pure dominant, parent 2 is pure recessive. So he have crossed the organism and he have got the first generation. First generation is known as F1 generation. F1 means actually filial. F1 actually means filial. Filial actually means son or daughter. It is bus. That is the meaning of the filial. So in his first generation, he got a heterozygous dominant. First generation, he have gotten heterozygous dominant organism. So I will say the experiment. He have taken a parent generation P1, P2. Consider P1 as a male and consider P2 as a female. The P1 in their cells, right? In their cells, they are tall. They are having a tall genotype. Here they are having a dwarf genotype. Okay, it is tall. It is dwarf here. But in their first generation, the organism which he got was tall. This is homozygous tall. This is homozygous dwarf. This is the dominant condition. It is the recessive condition. So while he conducted his experiment, he got a tall plant. So he was, you just understand, he was amazed. He was shocked. Because there were two parents with two different two different forms. One parent was tall and one parent was dwarf. But in the progeny, in the first progeny means in the F1 generation, he got every tall. Every organism was tall. F1, every plant was tall. He was shocked that his mother's, that means the mother plant's TT or the dwarfness was missing in the F1 generation. Why there is a missing? In order to understand the missing, he just conducted another experiment in monohybrid cross, which is the selfing of the F1 generation, to understand what will be the condition of F2 generation. F2 means the next coming generation. In the F1 generation, only one of the parents' characters was shown in the result, and he was shocked because the other parent's character was missing. Even though the cross was between two different parents, only one parent's character was shown. So in order to understand it, he just conducted the next experiment. Next experiment means just selfing the F1 generation. We know that F1 generation genotype is TT. He just self. Then what is selfing? So we are going to the next part of this experiment. Is about the selfing of the plant. Sorry. So you just check here. Here TT, right? TT means the gamete formed will be capital T. Here the gamete formed will be small t. We have crossed it, thus TT. I think there is no need to say these types of basic things because you have a basic understanding. I think you have a basic understanding about these things, right? So I also have given a question in the last lecture, how to find the different types of gametes. If the gametes are homozygous, then 2 raised to 0. 2 raised to 0 is 1. Only one type of gamete is formed. Here the only one type of gamete, which is capital T. Here also one type of gamete, which is small t. If you didn't understand this, please go back to the lecture and please understand it, because it is the basic thing, and this is the basic thing which you will be applying it into everywhere. So just have a basic understanding about the experiment. So we are going to the next part of the experiment, which was a selfing of the F1 generation. So F1 generation, he has self. So just consider this as a male or female. Just consider as a male. Yeah, I will just, this is according to your wish, right? Just considering it as a female, because I have given the priority to male in the last experiment, right? So I will give the priority to female. So I am conducting an experiment. F1 have been self. Self means the same genotype plan has been self with the other, or the same progenies have been self. I have taken one plant from the F1 generation and the second plant from the F1 generation, and I am crossing them. Crossing means pollination, right? I think you understand these type of things. So I am just pollinating them, because we are doing it in plants. So I am just pollinating it. I will say how we pollinate, what is certain, how he have done certain procedures to pollinate, like emasculation later. So you have just understand genetics part now. So I am taking a same F1 and a same F1, and I am selfing it. So just I am saying some mathematics here. Look here, TT, right? Here it is heterogametes TT. I know I have said that 2 raised to n. N is the number of heterogametes. Here there is one heterogametes. So 2 raised to 1, that is equal to 2. That means here 2 types of gametes will be formed. What are the 2 types of gamete? The 2 types of gamete will be TT and capital T and small t. Here also capital T and small t. You know how these gametes are formed? These are formed by the meiosis. In an organism, in a sexually reproducing organism, mostly, or in a sexually reproducing organism, the gametes are formed by the process of meiosis. You should understand the part of meiosis later, but here just understand, here 2 types of gametes will be formed, capital T, small t, capital T and small t. These are the, here 2 types I am saying, not the number may be very large or very small. I am saying about the types. So I am crossing it like T with this T and T with this T, small t with this capital T and small t with this capital T. How there is a particular sequence? Because it is according to meiosis. I have said it in the first lecture. It is according to meiosis. The chromosome is having AB here, and the same pair of homologous chromosome is having AB here. It will be separated to form 2 cells. Thus there is a separation of gametes. This is how gametes are formed. So just check here. This is the genotypes. Total number of genotypes is 4. But how many types? Total number of genotypes is 4. How many types of genotypes? Or total 4 progenies. Types of genotype is actually 3. What are the types of genotypes? Capital T, capital T, capital T, small t and capital T, small t, small t. These are the 3 types. This one, this one, this one. Right, there are 3 types. And what are the type of genotypes? Only 2 type of genotypes form, tall and dwarf. So I am saying some of the inference, something we have understood from this cross. There is a new genotype here formed. Actually from the, sorry, there is no new genotype, there is a phenotype formed, not a new, a phenotype of the mother in the first mother. That means this gametes grandmother, we can say, right? Grandmother was having the phenotype dwarf. That dwarf is re-expressed here. So just understood it. You just understand these things. In the first F1 generation, one of the form of the character was missing, but in the F2 generation, this is the F2 generation. In the F2 generation, the character have reappeared, which means that even though the character was present in the gametes, but it was not able to express, right? That is the thing. That is the conclusion we should make it from here. You just understood. In the first generation, the character was missing, the form was missing, dwarf was missing. But in the next generation, when we self it, he have understood the character have reappeared, which means that we can say we can just conclude two laws from here. One is that the character have been present in the cell, it has been reappeared. Why it was not able to appear in the F1 generation? It was because of the presence of the other dominant character which masks this character or which conceals the character, so it was not able to express in the F1 generation. But when we self the F1 generation to form the F2 generation, the character reappeared. So there is certain segregation, or there is certain process happening. By concluding this result and by concluding it mathematically, Mendel said that some factors have been transferred from the parents to the offspring, and these factors have two different forms. If the two different forms are coming in a heterozygote form, then the dominant form can mask or conceal the recessive form. Thus, if they mask, it will not be able to seen in the F1 generation. Or if it is in the heterozygote form, it can be seen. This was the conclusion which Mendel have made from his monohybrid pods. So if we check the result, number of phenotypes, I have said number of phenotypes, two types of phenotypes, tall and dwarf, right? These are the two types of genotypes. Tall and dwarf are the number of genotypes, right? Then the phenotypic ratio is 3:1. What is phenotypic ratio? 12:1 dwarf, right? How many tall was formed? 3 tall, right? 3 tall was formed, so 3. Next, dwarf, 1 dwarf, so 3/3:1 or 3/1. Tall by dwarf is equal to 3/1, right? Next, number of genotype. What is number of genotype? I have said how many type of genotype was formed. Here one genotype, here one genotype, here one genotype, or here one genotype, here one genotype, here one genotype. So 3 types of genotypes were formed, and the ratio is that 1:2:1. If you write it according to this, okay. So tall is to dwarf. Tall is to dwarf. Okay, that is the case. So how many tall was formed? 3 tall with 1 dwarf. 3:1 ratio. That is the case. So I think you understand it very clearly. So there are many. It is very interesting. You know, this chapter is very interesting because if you understand the basics of this chapter, it is very interesting for your studies, and you can score a good mark in your NEET examination. As I said, 13% of questions are being asked from biology part from your genetics. Genetics means this unit, inheritance unit. Okay. So there is also one thing to be discussed, which is the back rows. So we know the number of progeny is 4, right? 4 types of progeny, 4 number, not type, 4 number of progeny will be formed because 2 parents are involved here, right? With 2 parents are involved, only 2 parents can be involved, and all. But I am saying number of gametes. So that is the condition. So I will be saying some equations regarding this. Okay, number of phenotypes, you can write it in your notebook. Just write number of phenotypes is equal to 2 raised to n. Okay, and number of genotypes is equal to 3 raised to n. Number of phenotype is equal to 4 raised, sorry, number of progeny is equal to 4 raised to n. This right, number of phenotype is equal to 2 raised to n, number of genotype is equal to 3 raised to n, and number of progeny is equal to 4 raised to n. Just write this. And where n is equal to now n is actually the gene how many number of gene taken. For example, in a monohybrid cross, n is equal to 1. In a dihybrid cross, n is equal to 2. And a trihybrid cross, n is equal to 3. Actually, it is not needed. It is for your basic understanding. You just check. If we are doing a tetrahybrid cross or we are doing a trihybrid cross, if someone is asking, then what is the number of progeny? We can't calculate it or we can write it down. Just say that it is 4:3. 64 progeny will be formed. In a dihybrid cross, 16 progeny will be formed. How 4:2? Is the number, right? And how many genotypes? It is very difficult to write the entire genotype. So we can just say the number as it is 3n genotypes will be formed. Types of genotypes. In monohybrid cross, 3n genotype will be 3:1 genotype will be formed. Yes, it is correct. 3:1, capital D, capital D, capital T, small t, small t, small t. Yeah, 3 types of genotypes are formed, and number of this can be written. Okay, this is for your ease of understanding for your entrance questioning. We will do the questions very easily. Next is about the back cross. So, what is back cross? Just it is a small topic. What is a back cross? Back cross is the crossing of the F1 individual with any of their parent. Just write it down. Back cross is the crossing of the F1 generation with any of their parents. So we can divide the back cross into 2 types. Back cross can be divided into 2 types. First is test cross. Second is out cross. So what is test cross? Test cross means crossing of the F1 generation with their recessive parent. That is test cross. What is out cross? Crossing of their parent F1 generation with their dominant parent or a homozygous dominant. Yes, it is homozygous dominant according to Mendel's experiment, right? It is according to Mendel's experiment we are seeing, because he have taken homozygous forms of parents in their parent generation cross, right? Parent generation. So what is back cross? It is the crossing of the F1 generation with any of their parent. F1 into any of their parent, either AA or small a, right? So this is the case of back cross. So if you just note the name, it is written as test cross and out cross. Why it is given as test cross? You know, in F1 generation we will get an heterozygous gamete, right? F1 generation. So usually test crosses are done for any organism to understand whether the organism is in the heterozygous form. If the organism is in the heterozygous form, then the ratio will be 1:1. Then the ratio of the progeny will be 1:1. Yes, we will do the cross right now. Okay, I am taking the F1 generation. F1 generation is in heterozygous form. Okay, AA into, I am doing the test cross, small a. Here how many types of gametes will be formed? 2 types of gametes. This and this, right? Here only 1 type of gamete will be formed. So even though there are 2 number of gametes, but there is no use because everything will be equal. So we will write koi baat nahi, I will be writing 2 also. So I am giving the cross. Here AA will be formed, right? AA. I am writing it here, AA. Here the next one, AA. Here AA. A. Okay, what is its genotype? Just consider it as tall, dwarf. So here it will be tall, dwarf. So what is its phenotypic ratio? I am taking tall is to dwarf. Then the phenotypic ratio is 2:2. or we can write it as 1:1. Yes, dividing it by 2, right? 1:1. 1:2. So what did I say? If the test cross ratio is 1:1, then we should understand that the one of the parent are heterozygous. Sorry, the organism is heterozygous. Yes, if the phenotypic ratio is 1:1, then we should understand that the organism is heterozygous. Why did we test cross? We did test cross to understand whether the organism is homozygous or heterozygous. If the organism is heterozygous, then we should get the ratio as 1:1. So what will happen if the organism is in the heterozygous form? What is heterozygous form means? Tall. Yeah, the organism is tall, but it is in its heterozygous form. Here also the organism was in tall, but earlier it was heterozygous. So yeah, just if we look at an organism, we can't predict, we can't say it is in the genotype is homozygous or heterozygous. So in order to confirm it, we should do a test cross. So in case of a homozygous organism, I'm taking capital D as tall, and it has a cross between two parents. So here capital A, capital small a, capital A small a, capital A small a, capital A small. So all the organism here will be tall. So there is no ratio. There is no need of ratio. There will be no ratio, right? All the organism 1:1. Sorry, no 1:1. 1. There is no ratio because there is only one type of phenotype. If there is two types, then there is the need of ratio. So here there is only one type of organism. So we can 100% confirm in Mendel's cross that this is a homozygous parent, homozygous organism. This is the case of back cross and test cross. Back cross, there is no need. Actually, we should understand test cross. So the main portion is completed. Now we will be dealing about the extension of Mendelism. Actually, extension of Mendelism. Actually, in your textbook, after monohybrid cross, the codominance and incomplete dominance is shown. That is the reason why I am taking it accordingly. Right? There is actually, it is not in the proper order for me, but I am taking according to the textbook. Okay. Next, we are going to deal with the extension of the Mendelism. In extension of Mendelism, we will be dealing with three phenomena, or actually two phenomena: incomplete dominance and codominance. So if we go to the index page, the content we need to complete will be Mendelism. Okay, extension of Mendelism. In extension of Mendelism, we have completed all these things, and next is the incomplete dominance. Before going to the dominance, incomplete and codominance, let us discuss a small question. This is the question. The question is: a pure tall plant with yellow seed is crossed with a pure dwarf plant with green seeds. In the F1 progeny, they are having a tall with the yellow seeds. Okay. If F1 progeny is backcrossed with a recessive parent, then find out the phenotypic ratio. So the question is here is about backcrossing. We know that what is backcrossing. Backcrossing is the crossing of the F1 individual with any of the parent. That is known as backcrossing. Here it is backcrossed with the recessive parent. I also mentioned that if the F1 generation is backcrossed with the recessive parent, then it is known as test cross. So here a test cross is done. So let us do the question. A pure tall plant with a yellow seed. We know that it is done according to Mendelian crossing. Here two genes are taken, right? What are the two genes? The gene for height and also gene for color of the seed is taken. So it is a two gene crossing. If it is one gene crossing, it is known as monohybrid cross. If it is a two gene crossing, it is known as dihybrid cross. So here dihybrid cross is done and F1 is dihybrid cross is done and F1 is crossed with their recessive parent. So here a dihybrid cross is in a test cross in dihybrid cross, we can say that. Okay. So a pure tall plant means capital T Yellow seeds mean capital Y, capital Y. Y is given for yellow color. And parent 2 is having a pure dwarf. So small t, small t, small t. And green seeds means green seed is the recessive form of yellow seeds. So YY. So they have been crossed. We know that here only one type of gamete will be formed because 2 raised to 0. Anything raised to 0 is 1. So here one type of gamete will be formed and the gamete is T capital T capital Y. Capital Y. Capital Y. So here small t and small y. So if we cross this, we will be getting capital T, small t, capital Y, and sorry, capital Y and small y. So we will be getting a plant. Here it is given having a toll with yellow seeds. This is the condition. Now we are going to test cross it. So we will be test crossing it right now. What is test crossing? The crossing with its recessive parent. So we will be test crossing. Recessive parent genotype will be small t and small y. So if we cross here, we know that how much gametes will be formed. How much types? So we know that 2 raised to 2. How 2 raised to 2? Because there are 2 heterozygous conditions. So 2 raised to 2, 4 different types of gametes will be formed. 4 different types of gametes. What are the 4 different types of gametes? Capital T, capital Y, capital T, small y, and small t, capital Y, small t, and small y. Here both of them are in homozygous condition, so 2 raised to 0, only one type of gamete will be formed, T, Y. So if we cross here, cross these gametes, we will be getting, I will be writing the F2 here, capital Ti. I am crossing first this with this. So capital T, small t, and capital T, capital Y, small y. Next, this with this, small t, small t, y. I will say it's capital T, small t, capital Y, small y. You can write the cross, small t, capital Y, small y, and capital T, small y, small y, small t, small y, small y. So actually, if we check its phenotype, the phenotype will be having 2:2 ratio. Sorry, 1:1 ratio. Why? Because every genotype here every phenotype is different here. So the ratio is 1:1. Please check the phenotypic ratio. Then we will be getting 4 phenotypes here. And the ratio will be 1:1. So actually, we can confirm that if the test cross ratio is 1:1, then we can confirm that the F1 generation or the parent which we used, the F1 generation which we used was in heterozygous condition. I earlier mentioned that if in test cross ratio, if the test cross ratio phenotypic ratio is 1:1, then we can confirm that the F1 generation was in heterozygous condition. This is the thing which I was going to mention. So this is the question. Next, we are going to Mendelism, extension of Mendelism. Before going to extension of Mendelism, we need to understand something known as multiple allelism. What is multiple allelism? We know that in a human or in a diploid organism, a gene will be having two allelic forms. In a diploid organism, a gene will be having two allelic forms. Example, in case of tallness, in Mendel's experiment, we were having capital T and small t, which codes for tall and dwarf. That is the condition in a diploid organism. But for a gene, in a population, a gene may have more than two allelic forms to infinity allelic forms. That is the condition. In a diploid organism, gene will be having two alternative forms or alleles, but in a population, a gene may have more than two allelic forms to infinity. A good example of multiple allelism is the human blood group system, or the ABO blood group system is a best example of multiple allelism. So I have said two things, please write it. In a diploid organism, a gene may have only two forms, or a gene may have two alleles. But in case of a population, a gene may have more than one allele to infinity allele. A good example of multiple allelism is ABO blood group system. How ABO blood group system shows multiple allelism? For example, in ABO blood group system, we know that there are three blood groups: A blood group, B blood group, and C blood group. So, ABO, in ABO system, a gene may have three allelic forms. What are the allelic forms? IA, which codes for B and I. These two are the dominant form and this is the recessive form. Okay, these are the three alleles of ABO blood, of a blood group, which codes for a blood. So blood is the phenotype here. I actually said that what is a phenotype? A phenotype is a characteristic of an organism which may be its physical character, biochemical character, behavior, etc. This is actually the definition of phenotype. So here a phenotype is blood group, not the color of the blood. It's actually the blood group, right? So these are the three alleles in ABO blood group system. So we can write the combination here, right? So let me say that I am writing the combination. If IA is present in a diploid organism, we know that in a population three alleles may exist, but in case of a particular organism only two forms may exist. We know that if an organism is diploid, then two forms may exist. There are certain organisms which have triploid, tetraploid, certain plants, right? They are having these types of condition, or the condition may be also can arise due to some mutation also. So the thing is that in an organism, in a diploid organism, in these three alleles, only two alleles may be present. So if the two alleles are the dominant form of A, then it will be A blood group. So if one of the allele is A and one of the allele is recessive or O, then also it will be A. So there are two chances for A blood group. If a diploid organism is having IB, then it will be having a B blood group. Okay. Next, if the organism is having IB, the organism is having IB and I, it will be having B blood group. The organism is having both the I, then it will code for O blood group. So this is the case of multiple allelism, presence of more than one allele. So for doing questions, we have certain equations. It is for your ease of questioning, answering. So I will give an equation that in multiple allelism, to find the number of genotype, the equation is equal to n into n plus 1 by 2 to find the number of genotypes. Okay. To find the number of phenotype heterozygotes, it is n into n minus 1 by 2. To find the number of homozygotes, it is equal to n. This is just for basic understanding to do the questions. For example, here I will take the example of this blood group system. In this blood group system, we have 3 alleles. So the number of genotypes will be n is equal to number of allele. What is the case here? 3 into 3 plus 1 by 2. 3 into 4 by 2. 12 by 2 is equal to 6. Actually, there are 6 genotypic condition. Yeah. What are the 6 genotypic condition? 1, 2, 3. 4, 5. There is also one genotypic condition which I forgot to write. IA, IB, which codes for AB blood group. Right? AB blood group is also present. I forgot it to write. So there are 6 genotypes present. 6 different genotypes. So to find the number of genotypes, we know that n is equal to n plus 1 by 2. To find the number of heterozygotes, it is n into n minus 1 by 2. To find the number of homozygotes, it is n. We will do the questions later. First, we will go to the concept. So this is the concept of multiple allelism. The presence of more than one allele in a population is known as multiple allelism. That is according to the gene. So next we are going to discuss about the incomplete dominance. What do you mean by incomplete dominance? It is actually the extension of Mendelism. What is incomplete dominance means? Right? In incomplete dominance, so first of all, we will do a cross. Incomplete dominance is also known as semi-dominance or intermediate dominance. Why it is known as semi-dominance or intermediate dominance? Because we will understand it from here, from this cross. So in a cross, I took a red homozygous plant and also a white homozygous plant. The character is color, red and white. So I have done a cross between red. So here the gamete will be R. I think there is no need to write it every time. You know it. So if I cross it, the gamete should be RR. The gamete or the zygote, sorry, the zygote is RR. It has fused to form a zygote. Or the plant is RR. So you just look into the cross. Actually, it should be red, right? The dominant form is red. So actually in a heterozygous form, the dominant form should be red. But here, so in this condition, it have become pink in color. Actually, the color pink was not even in the parent condition. So this is known as incomplete dominance. The color is actually the intermediate of red and white, pink color. So this is known as incomplete dominance or semi-dominance. Actually, the dominant form of an allele can't express fully, or the dominant form of an allele can't mask the recessive allele completely. It is known as incomplete dominance. So this is the F1 generation, this is the condition of filial 1 generation. If we do the cross, if I am selfing RR, if I am selfing RR into RR, in the F2 generation, what will be the, it's actually like monohybrid cross, RR, RR, RR and RR. So here it will become red, these two will become pink and this will become white. So this is the condition. You just check. In F2 generation, we know that the parent genotype or parent phenotype have came back. So the phenotypic ratio will be 1:2:1. Genotypic ratio is normal, 1:2:1. So this is known as incomplete dominance. You know, here each genotype has its own distinguishable phenotype. But in case of monohybrid cross, of normal monohybrid cross, the condition was not like this. Three genotype were having the same phenotype, 3:1. That was the phenotypic ratio. Tall is to dark, 3:1. But in case of incomplete dominance, both the phenotypic and genotypic ratio are equal. What is incomplete dominance? In incomplete dominance, it is also known as semi-dominance or intermediate dominance. In intermediate dominance, the dominant form of the allele can't express or conceal the recessive form of an allele, so an intermediate form will be obtained. This is case of incomplete dominance. Usually it is seen in an organism, seen in a plant, Mirabilis dilapa, family Nyctaginaceae. Okay, this is the case of incomplete dominance. I think you understood it. So there are certain conclusion. It is known as semi-dominance, intermediate dominance. One allele is not completely dominant over the other allele. Total gene product is the intermediate of the dominant and the recessive allele. Every genotype has a distinguishable phenotype. Please write it down. Next is about the codominance. Codominance. We are taking codominance. Codominance is usually, we can give an example of ABO blood group system. We know that in ABO blood group system, there are 3 alleles in a population. What are the 3 alleles? A, B, O, or A, B, I, where I is the recessive form. So here in the cross, we are taking the parent which is homozygous dominant, 2 dominant forms of parents, and we are crossing it and we are getting I, A, I, B. So if it is IA, the blood group IA, IA, okay, homozygous. It is also homozygous. IB, IB. So here only one type of gamete will be formed, and if we cross, we are getting a different type of blood group. Parent was A blood group, the other parent was having B blood group, but in the progeny, every progeny will be, it means every son and daughter will be having AB blood group system. AB blood group. If the parent is A, homozygous, and it is actually in homozygous condition. In heterozygous condition, progeny may be different, having different blood group. All progeny may be having different. But if the parent are in homozygous condition, A blood group, B blood group, and if we are crossing it, we will be getting every progeny which will be having AB blood group. So what happens if we self it? Actually, the result we are looking in F2 generation. So this is the condition of ABO blood group system, and it is an example of codominance. Okay, so we are going to selfing it and we need to find the F2 generation. So I am selfing it. Actually, the result, every result is the result of the F2 generation. So if I self here, the number of gametes will be IA and IB. Heterozygous condition, so 2:1, 2 types of gametes will be formed. Here also 2 types of gamete will be formed, IA, IB. So the result of the F2 generation, F2 cross will be, the progeny will be I will write AA, IAA, IAA, okay, then IA, IB, then IB, IA, also sorry for my handwriting. Okay, writing A, A, then AB, AB, and also BB, BB. So if you check here, we are getting four progenies. So it will form A blood group, it will form AB blood group, it will form AB blood group, and it will also form B blood group. So here also the phenotypic ratio and genotypic ratio will be equal. What will be the phenotypic ratio? 1:2:1. A is to AB is to B. Genotypic ratio is 1:2:1. That is normal. So in case of codominance and in case of incomplete dominance, both of them phenotypic ratio and genotypic ratio are equal. If in question they say that both the genotypic and phenotypic ratio is equal, we need to understand that it is the question is from either the question is in the extension of Mendelism, from the extension of Mendelism. So this is the case of codominance. You will understand it very clearly if after we do the questions. Doing question is very important to understand the concept much deeply, and also you should read your textbook. Okay, actually the codominance and incomplete dominance are a part of gene interaction, right? There are actually two types of gene interaction in within a particular alleles of a gene, and also the other condition is that gene interaction between two different genes. The gene interaction with two different genes is known as intergenic gene interaction, and the gene interaction within the alleles of a particular gene is known as intragenic gene interaction. Codominance and semidominance comes under intragenic gene interaction. It means both of the allelic form of the gene gets interacted, they interact and form some extraordinary things, which we discussed in codominance and semidominance. So in codominance, what happens? Codominance means both of the dominant form of a gene are expressed equally. So here in AB blood group system, we will be getting ABO blood group system. If A and B are expressing together, we will be getting a new blood group which is AB. So that is the thing to be remembered. So I have written some conclusion also. In codominance, the heterozygotes exhibit the phenotype of both the homozygote. Okay, in codominance, the heterozygote will exhibit because they are expressed equally. So this is all about your part 2 lecture. This was the things we discussed here in part 2. I think this was easy for you. Here please correct this spelling, right? So I think it was very easy for you. So we will be seeing in the next part 3, where we will be discussing the dihybrid cross. Actually we have discussed it. What is a dihybrid cross? Cross with when we take two different character and cross, when we are only considering two different characters. So I hope you like my lecture, and we will see in the next lecture. Thank you. Please subscribe my channel and please like. Please mention your valuable comments, the things I should clarify with more details, things I should make much more. Thank you, and thank you.